Okay, is this working? Hi, welcome to the last seminar of this semester. Thank you for joining. Today we have the great pleasure of having Andrea Paz here with us. She did her master's and her bachelor's and master's in Bogota, and then she moved to New York for her PhD. She worked with Ana Carnaval, studying the Atlantic forest in Brazil mostly. She's gonna say more about it. Then she moved here to Zurich. She's working at ETH as a postdoc in Crowther's group. And the good news is that she's soon moving to Canada for an assistant professorship. And she's gonna tell us more about what she has been doing, her plans for the future, and how passionate she is about diversity patterns and the process that drive and maintain these patterns. So the floor is yours. Thank you, Andrea.
Thank you. I think this is working. Thank you, Elisa, for inviting me. I'm really happy to be here and really happy to be able to tell you a little bit about what I've been doing here, but also when I was studying the Atlantic forest, which I think was part of why Elisa invited me. So it's going to be sort of the biggest part of my talk for the Brazilians a little bit. I've been really interested like throughout my career in studying the distribution of biological diversity. And I'm sorry that I was told I cannot move further away from the desk or it's not on camera. So I'm just gonna try to do this. Yeah, so I started my career really curious about diversity patterns and about understanding why some places had so much diversity and other places had so little diversity. And being born in one of the most diverse countries in the world, only rivaled by Brazil, I was born in Colombia. I was really curious about diversity overall. And I have been interested in studying this at 3 levels or 3 scales. The distribution of populations and species at the landscape scale and how the relationship with environments is, then the spatial distribution of biological diversity and their drivers at a regional scale, and that's gonna be mainly the Atlantic forest, and more recently studying the distribution of biological communities at a global scale. So just a brief overview of how I started. I started working with frogs. I started as a herpetologist. I started very interested in understanding how these frogs were distributed across different landscapes. And my first ever sort of big research project was on the frogs of Central America and trying to understand how they were distributed across common landscapes and what explained the differences in their genetic divergence over this common landscape. And so I'm just going to show you one example. I guess this doesn't work like this. Oh, this one. Yeah. So I'm going to show you this comparison. So those are 2 mountain tops in Panama, but then I also did this comparison. So for those first 2 mountain tops, they shared about 30 species of frogs. And for all of those species, I got DNA barcoding data. So just like small pieces of DNA. And tried to explore whether they were diverging at the same time. So was it just this valley here that explained how they were diverging genetically, or was it something else? Was it something about the frogs themselves? Was it something that was making them different? So was it the organisms? Was it their life history? Was it their traits? Was it just climate, just the fact that it was a valley? Or was it somehow the interaction between what those organisms were, their shape, their life history, and the climate they were experiencing? It was at that time that I started working with species distribution models, and I know that you know a lot about this here, so I'm not going to talk much about it. I was sort of using presence localities for this species of frogs, combining them with climate, creating maps where I would know what the most suitable habitat was in red and what the less suitable habitat was in blue, and projecting this to the past in this case. So in that case, to the last glacial maximum. Just remember, this happened a long time ago. And obtaining measures of resistance of the landscape. So how resistant is the landscape to the frog crossing? What I found was that for that comparison that I was showing you, we had 3 divergence events. So those 30 frogs were basically divided in 3 groups that were diverging at different times. And what really explained those different divergence times was landscape resistance, both in the last glacial maximum and in the present. So following that idea, I started working in the Atlantic forest. So I went to Brazil, sort of started studying the coastal forest of Brazil, and found these two glass frogs, Vitrorana aurignata and Uranoscopa, and they had sort of the same idea of the story that I was telling you from Central America. They were distributed sort of in the same area, and yet they had different divergences. So try to explain how and why are they having these different divergences and other things in common. Same idea, I try to create models and now get an idea of landscape resistance across time. So it's just not one time period, not just the last glacial maximum, but just across 120,000 years. What were the areas that were harder for species to cross? What I found was that just like with the Central American frogs, landscape resistance really explained how those populations were structured. Landscape resistance across time. So frogs just were a really nice model to look at the interplay between environments and species traits. They're very related to their environments. We love them, they're really cute, they're easy to catch. So I continued working with those models and frogs for a while, trying to look a little bit further. So if in fact environments are helping explain their distributions, can we predict and generate hypotheses about their past and future distributions given climate change? So starting with looking at the past, we looked at 7 highland species and 7 lowland species of frogs in Colombia, in the Andes of Colombia. And so this was done by some of my undergraduate students, and predicted how their elevation was going to change, their elevational distribution was going to change in the past. What we found is that you can see in blue the present elevation, and red and yellow, I guess, is their projected elevational distribution in the last glacial maximum. So what we found was that high elevation species were predicted to go downward, whereas lowland elevation species were predicted to stay put. If we look at their area change, the area of the highland species was increasing, so they were going down and expanding, whereas the area of the lowland species increased only if they were associated to xeric habitats. So it was more related to precipitation. If we look at the future, sorry, that's what happens when you go to PowerPoint. So looking at the future, again, for some species from the Andes, what we saw was that for most species, they're predicted to be increasing their elevational range. So here, black is the present. I can use this. And the colored ones are the future. So that's just one example. We did it for 28 species and found that over 63% of our high elevation species are predicted to be pushed upward and reduce their area of distribution. And even though this is just models, this phenomenon has started to be reported for several species of frogs in the Andes of Ecuador. So not Colombia, but just monitoring efforts are different in different countries. So it seems like these models could help us do some predictions. And in that sense, I got involved with this project to try to make species distribution models more available, easier to teach, easier to replicate. And so I just wanted to briefly mention WALLACE as the multi-institutional, multi-country collaboration that we have to create a GUI app that is easy to use, but still instructive, and still people can sort of make their decisions based on the literature and based on thinking of some of these decisions. We've used it for teaching a lot, but also we've started to use it for conservation applications. So trying to bring this into the places where conservation efforts are needed, to work with conservation practitioners, to work with the governments, so that they can do the models themselves in the most correct way possible and apply it to their different countries. So we have a pilot in Colombia where we're working with the Humboldt Institute, and they're integrating this with their platform, trying to map all the species in the country, which has been an interesting experience. So if you're interested in WALLACE at all, we can talk about it later. So to close this first part, the interaction between organisms and environments are drivers of population structure, and we can use this association of the species with the environments to predict their present distributions, but also their past and future. And I am now very interested in understanding a little bit more about the mechanisms that are driving this in amphibians, sort of a little bit more about the physiology, what's happened to the individuals. And I'm not going to show that in this talk, but I'm happy to talk about it later. But I'm starting to get some results from experimental work. But that means doing a lot of work in the field. And that means it takes a lot of time, and that means it is limited in its geographical scope. And it's impossible for me to cover an entire biome, impossible even more to cover the entire globe. So to try to get at this question of how do we look at the spatial distribution of biological diversity at a regional scale, I had to stop with the collecting my own data and start collaborating with others and sort of go into this awesome ecosystem that has so much data and so many people working in it, because it allowed me to get access to data from different groups of species, plants, animals, and like a very good geographic coverage of different things. So I know that there's a lot of Brazilians here, but still just a brief overview of what the Atlantic forest biome is. That is the delimitation of the biome. That is definitely not what remains of the Atlantic forest, which is about 10% of what I'm showing there. It is a beautiful place. It has sort of all the landscapes you can imagine. It goes from sea level to the mountains. It has the dry forest. It has the rainforest. It's really a good place to look at environmental heterogeneity and try to quantify diversity. And then we get at the question of how do we quantify diversity in a place like this? Is it just the number of species? Is it how different the species are? Is it how they are related? And in that sense, for the Atlantic forest, I started looking at different dimensions of biodiversity. So species diversity, yes, how many species? But also functional diversity, what are their ecological traits? And phylogenetic diversity, how the species are related. And I'm going to start with the frogs and then move on to different organisms. These are hyalid frogs, the tree frogs. They are very common in the Atlantic forest, but most of all, they're very variable. Some of them are really big. They can be as big as my hand, as my palm, a little bit bigger. Some of them are really tiny, just like a tip of a finger. So you have all this trait variation that allowed us to look a little bit more at how functional diversity was distributed across the forest. So to try to map this, I looked at the richness, the species number, the evolutionary history, and the ecological diversity of hyalid frogs across the forest, having a per pixel measurement. That was based on, you guessed it, species distribution models, just like I was showing before. If you look at the results, so you have high diversity in red, low diversity in blue. We have sort of similar results for species richness, phylogenetic diversity, and functional richness, with functional richness being the one that is slightly different from the others. I don't know why this is now red, Nadia, and it doesn't do the... Anyway. Slightly different, but it's hard to say where exactly we find the differences. Mostly we know that the Atlantic forest has higher richness of all of these measurements in the coastal forest, in the Serra do Mar, and the Bahia coastal forest, and in the case of functional richness, a little bit inland. So how do we look at the differences? We can regress these things onto each other and look at the residuals. And what the expectation would be here is that this is just for functional diversity. If we have higher functional diversity than expected, higher diversity of all these traits, then we think maybe this is the product of competition, maybe this is the product of contact with other biomes. The Atlantic forest has contact with the Amazon, the Cerrado. If we have less functional diversity, so if things are looking more alike than they should, then it's probably the effect of environmental filtering. So as environments get difficult, things sort of look like each other to try to survive those environments. It will be a similar idea for phylogenetic diversity. So what happens with the Atlantic forest is we have a little bit of everything, as I just described it. So the north of the Atlantic forest, it's mostly lowland, it's mostly almost tropical. It is a nice weather for the species. I don't know. It's not very hard. So the expectation would be we have more morphological diversity, more traits, more reproductive modes than expected. It's like there's space for more things. As we go south, the south is no longer tropical, it's seasonal, it gets snow. It also has a little bit more of an elevational variation. It's just overall a harder climate for a frog to survive. And here in the middle, we have the Serra do Mar. So we have some of the biggest peaks, the highest peaks in the Atlantic forest. We have a big elevational difference. So we expect things to be changing with elevation instead of with latitude in the middle of the forest. So let's look at them individually. For the north, we found that functional richness residuals are indeed positive. So we have a lot of diversity for all these traits and reproductive modes. Phylogenetic diversity, on the other hand, is less than expected. So things are extremely related. So we have many and different traits, even though species are closely related. So they're being pushed to diverge. Potentially as a result of competition and contact that is driving trait diversity regardless of their relatedness. If we look at the south, these are the easy ones. The south, remember, is just hard. It's cold. It's difficult for the frogs. Functional richness residuals are negative. So we have less diversity than expected. Things are more similar to each other than we would expect just by chance. Phylogenetic diversity, on the other hand, is positive. So we have things that are not very related to each other and yet are very similar to each other. So the low trait richness, but species are not closely related. Strong environmental filtering can explain how things that are very divergent are still looking the same. They have to look the same to survive these environments. Looking at the middle, not exactly the middle, but just the Serra do Mar, with the incredible elevational variations, you can see it goes from 0 to over 2,500 meters above sea level. We can see that functional richness residuals are decreasing with elevation. So at low elevations, we have positive residuals, and at high elevations, we have negative residuals. So this is the same as the north at low elevations and the same as the south at low elevations. Looking at phylogenetic diversity, this pattern is even more dramatic. Very positive residuals at low elevations, very negative residuals at high elevations. So it replicates the south. Overall, trait diversity is reduced in the highlands, and species are more closely related in the highlands than in the lowlands. So it seems like environmental filtering in the highlands is homogenizing traits, but also filtering for closely related species. Whereas the lowlands have a diversity of processes happening. It's warmer, probably there's space for more competition. Again, it gets contact with the nearby biomes. So it could also be things are coming from the neighbors. So just to summarize, in the north, traits are diverse but closely related. In the south, traits are homogeneous, even though there is diversity of evolutionary history. And in the Serra do Mar, elevation is driving it, so elevation mimics latitudinal patterns for functional diversity, with lowlands looking like the north and highlands looking like the south. And we can talk about this when we have time for questions. I showed you what happens with the regressions. This is true using null model simulations also. It was just clearer if I show you the regressions. So I started thinking, okay, so environments are really important. Can we use them to predict patterns at the community level? So can I go beyond just like the species and predict the communities? And for this, in the Atlantic forest, we collected data for many, many groups of plants and animals for both species presences and phylogenetic information. Couldn't get trait data for all these groups. So what we had was a little bit of everything. Birds, some plants, some frogs, some butterflies, and those come from different groups that work in the Atlantic forest. You can see the credits to the pictures there, but I promise they're there. It's just getting cut. It's okay. And then I didn't do models in this case. We created polygons around those points, stacked the individual maps, and mapped species richness, phylogenetic diversity, and the residuals, just like we just saw for the frogs. So this is just the plants. And what you can see is that species richness and phylogenetic diversity look very much the same. So the first row, the second row, they're correlated as expected. So we look at the residuals. The residuals do have sort of different patterns of high and low, depending on which group of plants we're seeing. Basically, we see two big patterns. Some of the groups have higher diversity in the Cerrado Mar, just like I showed you for the high-lid frogs before. So that's the area that has a high elevational variation. Some of the groups have higher diversity, like the bignonias, in the north of the forest. If we look at the animals now, you can see some of the same patterns. So these tanagers still have higher diversity in the Cerrado Mar, but we find one more, which is some groups are just distributed across the forest, everywhere, just like those butterflies there. So can we predict this, even though they have very different patterns across the forest? We tested these variables. So they're related to temperature, precipitation, topography, but also past temperature and past precipitation. So variation in temperature and precipitation across time in the Atlantic forest. And found that using machine learning models, we can indeed predict richness and phylogenetic diversity very well. And what was maybe more exciting to me was we even can predict their mismatch. We can predict the residuals quite well for all the groups. Some better than others, but still it's good for all the groups. If we look at what variables are predicting this, it is mostly driven by climate. So you can see there in blue, things that are related to precipitation, current or past. In red, things that are related to temperature, current or past temperature. Topography had very little effect overall, which was exciting with the idea of projecting into the future, because then I could use only things that are changing. Everything that I just showed you was based on weather station data. WorldClim, CHELSA, but there is satellite information available from NASA and from other sources that gives us information on temperature and precipitation in a more sort of time informative way. We can use this and we can process it sort of in many ways. Most of it gets taken every 2 weeks, so we really could process this in different ways and look at timing. So I used some of the data available from NASA to get at bioclimatic variables, but annually. So that's what you're seeing in that GIF is just annual mean temperature changing from 2015 to 2020. But I did this for all the bioclimatic variables to have them for every year. Using this remote sensing data, we couldn't predict all the groups as well. But we could predict some of the groups, basically those that had bigger overall total distribution. So that meant one of the group of frogs, some hylids here, some of the plants, the tanagers, and the butterflies. So I kept going with those groups that were predicted well. To try to predict them today, but also predict what their richness and phylogenetic diversity would be in individual years that have already passed. So projecting this to 2015, 16, this should be 17, but it's cut, and 18. Yes, that's not exactly the diversity in every single year. Things are not changing this fast, but it gives you a trend. So we could compute which areas are predicted to be gaining or losing species over time. So for all the groups that I just showed you that we could predict pretty well, I'm showing you here maps where red means they're gaining species and blue means they're losing species. And again, this is species specific. It's idiosyncratic. Every group is losing and gaining species in different places. And this is just a model. And I know when I see your faces that you're not believing me with this model. Because it is a weird thing to say. But the Atlantic forest, I go back to, this is an amazing place because people have been monitoring the Atlantic forest for so long that we had some data to do some ground truthing. So I applied this model to data that was collected over the years for the forest in places that were monitored, for frogs and for epiphytes. Those were the two groups that I could find. And this is the actual observed loss and gain of species in those sites for those groups. So again, places that are in red are gaining species, and places that are in blue are losing species. And this is not the model. This is what's happening. This is the model. This is applying the model that I just showed you to the same data set. It's hard to tell. So here's the accuracy. All the ones that are in black are correctly predicted. The ones that are in dark gray are incorrectly predicted. And the ones that are in light gray, we just didn't have information for that area. So you can see, didn't realize it was so cut, but you can see there's a lot of black. We are predicting quite well. If I give you the numbers, for the frogs, we were correct for 76% of the pixels, which is statistically significant. For the epiphytes, we were correct for 61% of the pixels, which is not exactly statistically significant, but still, we were correct for 61% of the pixels. Still better than 50%. We're not just guessing. What's maybe more interesting is that the model is way better at predicting areas that are losing species than areas that are gaining species. And areas that are losing species are really the areas that we want to predict. We want to be able to know which areas are more at risk. And then those areas we can pinpoint and say, those are the areas that we need to go and survey more. Those are the areas that we need to think what to do with the species that are there. Because in this case, they're losing species just because of climate. We're not even talking human intervention here, which is pretty bad in the Atlantic forest. So these are areas that are at risk, whatever we do. So we can think about the species that are there. What should we do? So it is possible to monitor species from space, at least for some groups and dimensions of diversity. This means we can rerun this all the time with new climatic data, and we can give ideas of this is the trend for these different areas in the forest. And if this is the trend, then maybe these areas that are losing species, we should pay more attention to them. And we can do this mostly for groups of species that are broadly distributed, which, yeah, I would like to do it also for the ones that are not broadly distributed, but haven't been able to so far. At least for those groups that are broadly distributed, we can pinpoint the areas that are experiencing diversity loss. And then maybe the Brazilian government can do something, maybe now. When I presented this 4 years ago, I wasn't that sure that I was giving any kind of good information. Anyway, so going to the last part. After doing a lot with the Atlantic forest, and I'm still interested in looking at these patterns at the regional scale and at the drivers at the regional scale, and I would love to be able to apply this model to other areas. In particular, of course, I'm really interested in the Andean region, so would love to have data to test that, which I don't at this point. I started working in the distribution of biological communities at the global scale. And then this becomes even more difficult. There's no way I'm measuring frogs for the entire world. There is now a database out there that might be good enough. But when I got here, it turns out trees had a pretty good coverage. So I switched to trees to try to do sort of the same approach that I was showing you, but at the global scale. So can we map species richness, phylogenetic diversity, and functional diversity for trees at the global scale? And again, as expected, those measures are correlated with richness. So this doesn't work anymore. They're correlated with richness. So areas that are high diversity in red and areas that are low diversity in blue are mostly the same between all three dimensions of diversity, although you can see some areas that are a little bit divergent. But when we look at a different measure, when we look at divergence, so how different is a species in an area, then the pattern changes. It's still low divergence in blue and high divergence in red. And now this is not the map that you see all the time. This is not the map where the tropics are the biggest diversity. What we're seeing is that there's a hump-shaped pattern for both functional and phylogenetic divergence. That I can't point anymore. And so we have a latitudinal gradient of species richness that we all know. with species richness decreasing as we increase in latitude, but that's not what happens with divergence. Is it working now? No. With divergence, we have a peak at mid-latitudes. So both in the tropics and in the high latitudes, we have lower divergence in both phylogenetic and the functional dimensions, which is interesting. There's something different happening in. Thank you. Anyway, now I have stick. And now it's working again. Yep, here. Okay. So we have a hum shape, which has been established. And to predict these measures of diversity, again, I tried different things. It is still climate that best predicts not only richness, not only functional richness and phylogenetic richness, but also there's different patterns in divergence. So divergence would be this one divergence in phylogenetic dimension, and this one is divergence in the functional dimension. Sorry for the cutoff. It is mostly climate. It is different things in climate, but it's still climate that is predicting these differences, and it's still climate that is predicting this different shape in divergence. So this is at the community level. I was still interested in understanding whether this is happening because of some process at the species level. Is it the community? Is it the species? So we try to look at the relation between environments and plant species distribution at the species level. So this is Marco Barandon's thesis, his master's thesis. He just finished last year. This is classical Rappaport rule. As the latitudinal median increases, we're expecting the latitudinal range to increase. That's true for the trees of the northern hemisphere. So graph C here. That is kind of true for the trees of the northern hemisphere, for the herbaceous species of the northern hemisphere, although not completely. But if we go to the southern hemisphere, then that's completely not true. That pattern is inverted for all species that are outside of the tropics. As we increase the latitudinal median, the latitudinal range decreases. So the question we have was, is this related to environments? Again, is this related to the environmental breadth of every individual species? Again, for the northern hemisphere, it's sort of there, although the slope is not very steep, but it's still there. Very similar for the herbaceous species. But the pattern is broken for the southern hemisphere. It doesn't mimic what I was showing you before. It's mostly a flat line. So it's not related to the environmental breadth of individual species. So diversity measured as richness does have the strong latitudinal gradient. Functional phylogenetic divergence have a hump-shaped latitudinal pattern. And climate is the most important driver of these different patterns at the community level. But environmental breadth of individual species does not seem to predict this latitudinal range. So there's something happening more like community-wide rather than species level. So with this, I wanted to thank all the institutions that have been part of some of the things that I showed you here, and all the labs that have helped in different ways in doing these things. And I also wanted to take the chance to say, if you're interested in these topics, if you want to talk more about it, if you're thinking about graduate school, about sending your students somewhere, postdoc opportunities, I'm starting my lab in the spring 2024. And I'm very much looking forward to talking more about all the dimensions of diversity and all the scales in which we can measure this. Thank you.
Thank you, Andrea. This was very nice. And we're open for questions.
So I had a couple questions, and you probably thought of this, but you had these specific places where you had enough data from the literature for, I think it was for epiphytes and something else, right? And then you had all these groups that you didn't have enough information for, but that was at a different step. So are there some places where you have maybe not the ability to model, but the ability to say how many species there are? Because if there's correlations between the raw, like the real data, the observed data, then maybe you can sort of assume that these broader models do say something about the observed data. So you have
I think
a subset of observed data for
Yeah.
which you have broad scale data, and you can make this comparison. And you would like to make this comparison for everything, but you can't, because you couldn't model, I guess, with the remote sensing data, a bunch of those species.
Yeah.
But do you have information for those species groups in those local areas where you could sort of see if one could be a surrogate for the other?
Yeah, I sort of see what you mean. I mean, I could model them if they were just not very well predicted, and I did model them because I wanted to know how it looked. But yeah,
are they showing similar trends? So in those places, are the different groups, like that whole long list of groups, showing similar trends?
Let's just
Do
go
you
back.
have that information?
Do you mean, yeah. So yeah, for some of these groups that I couldn't model for, is that what you mean?
Yeah, I think it was later, but.
Yeah, I guess, yeah, you mean here, when I said I couldn't model.
Yeah, so you
those.
couldn't model all of these, but are these groups actually showing similar trends in your local sites where you have information?
Yeah, I see your point. Yes and no. I mean, just like I showed before that we have like 3 big trends in the Atlantic forest distribution, we also have some groups that behave the same, and like groups of groups that behave in the same way than others. So yes, we could use that to say one of the things that I didn't model behaves just like the hylids. So I'm going to say they're going to be losing species sort of inland here. But yeah, I don't trust it that much. I think we just need, maybe more data. We could definitely get more data at this point. Mostly those species are very endemic, especially these bromeliads. And so they're very restricted. Each individual species is very restrictive. So it's hard to get higher. But yeah, I guess we could find more. There's something.
Thank you very much for your talk. I have a question about the different temporal scales you used in the different studies. In the first case, you used past climate data back to the last glacial maximum, I think. And in the other study, you used LiDAR data back to the 2015 years, I think. So how did you define these different temporal scales or periods to predict this environmental filtering and then the community changes over time?
Yeah, so in the first things that I showed you, it was more looking at the genetic variation. And so the expectation there was that past climate would have a bigger impact on genetic variation than what you would see in climate right now. I did use the last glacial maximum, but also for the Atlantic forest, because we had more data, I used 120,000 years. So last glacial maximum was 21,000 years. So this went 100,000 years backwards. For the community one, it was trained on remote sensing data and projected on remote sensing data. So the dates are basically defined by when that satellite is flying. So MODIS started producing data in 2002 and has data up to 2020. And then after that, it's sort of less reliable, but it's still something there. So that's what I could have. That's just what the satellite has been flying.
I'm not really familiar with the South American continent, but in the Alps, we think about these 100 years to get the climax in the alpine grasslands, for example. So I was trying just to translate what can be expected in this Atlantic ecosystem, whether it would take, I don't know, a few decades to reach a new climax, for example, under environmental changes.
Yeah, I think definitely for gaining species, we need more time. And so this is just a train that it will probably gain species, but it's not actually gaining species right now. For losing species, it can happen quite fast, especially with extreme temperatures. So yeah.
Thank you.
Thanks for the talk. I have 2 questions. So first question is like for the species lose or gain in the South America. So what's the main reasons for the lose, for instance?
What's the main reason for what? Sorry, can you?
Main reason for the species lose when you point
For
out.
losing the species?
Yeah.
The main reason?
Yeah.
Well, in real life, the main reason for losing species right now in the Atlantic forest is deforestation. This is just like added to with the models. I'm showing that also climate is causing species loss, but that forest human impact would be the main reason right now.
Okay. So because you run the models the most using the climate, but you didn't include the human disturbance somehow, or?
Yeah, not for the predictions. We've included in something that I didn't show, but like this one. We looked at the environmental distribution of the remnants of the Atlantic forest, given human intervention, and showed that the remnants have a very good representation of the entire environmental distribution of the Atlantic forest. And so we think we're pretty good predicting environments because what's left of the forest is environmentally representing the whole forest. So if you want to look, oh, that's what you can see here. So if you want to know more about this, this is a paper from Jason Brown and Diversity Distributions in 2020, where we added sort of the human impact to exactly these maps. And the citation was there. It's just, we can't, yeah.
Yeah, and another maybe is because I didn't feel this from the same field, so maybe I ask just like very simple and naive question. So what's the functional diversity really stands for? Like what does that mean in real meaning somehow? If
well, it depends on what you're basing it on, right? So when I was showing you functional diversity for the hylid frogs, I guess no. Anyway, when I was showing you functional diversity here for these frogs, it is a compendium of both morphological data for these frogs. And morphological data, I mean, all measurements of size and legs, and along with a little bit of life history and reproductive modes, and some of their preferred habitats for the frogs. When I was showing you functional diversity for the trees at the end, then we're talking tree height, different measurements of leaf size, leaf weight. So it really depends on what you're using. Yeah.
Thanks for questions.
Thank you. I'll just pick up on this question. And for the plants, do you also have reproductive traits like seeds or
seed size,
reproductive mode?
Yeah, well, based on measurements. So like seed size, not like what disperses the seed or yeah, but yeah.
I'll just ask one more. I was curious with the near real-time modeling with the satellite data. Could you see a difference on how well you predict the gains and losses based on dispersal capacity of the organisms?
We try to include dispersal capacity. There's just not enough information for those groups. And within a single group of plants, we had like all these different dispersal modes that it was very hard to sort of summarize it as the group. So like as this is the dispersal capacity of all bignonias. That's not, it didn't work. Even with the sort of like only the hylids, so there's no dispersal capacity of the hylids because you have them, some of them are so big and some of them are so small. So it was hard to make it a group category. Yeah.
So you, I think in the middle study, you were looking at these possibilities of environmental filtering, say in the south or in the harsher environments. Did you consider sort of taking the next step and maybe defining a more restricted species pool where you consider those species that are then filtered and then ask whether you can additionally see some sort of even spacing or something like this that would be indicative of multiple mechanisms? Or
yeah, so I did it. I did a global species pool for the whole Atlantic forest, and then compared to the null models. And then I divided the Atlantic forest, how Anna had divided it in like north and south environmental spaces in that old paper. So then divided the species pools as north and south, and again did the simulations. It still shows that the south has very strong environmental filtering, and I might have the slides here. Let me see.
Oh, it's okay.
I think, yeah, I think they're here. You can see it. Yeah. So if you here, this is the results of the null model, and this will still be lower than expected with splitting north and south, and that will be higher than expected. So it's no longer the whole forest, but it's still south and north are very different. Yeah.
You showed that the annual mean temperature was the better predictor of the community changes over time. Was it? Did I got it right? Or
not annual mean temperature. So the different temperature. Yeah.
So it's interesting that from the LIDAR data, you see that the temperature gradient is correlating with the latitudinal gradient. On the other hand, some groups have a higher species richness in the north or in the middle or in the south. Did you see any differences in this pattern between taxonomic groups, like one that had different species richness in the south or in the middle or in the north regarding this mean temperature? Or?
Yeah, they definitely have different specific contributions of variables. I mean, it was what I showed you was an overall temperature on overall like this, but you can definitely see that different groups have different relative contributions of each one. The overall temperature and precipitation are the main drivers, but you can see, for example, that the bignonias here have a higher contribution of precipitation than temperature than other groups. So yeah, there's definitely variation that explains why some species are in some places and not in others.
It was somehow possible to disentangle environmental filtering from demographic processes somehow, the migration from north to south and so on.
Yeah, I mean, not with this approach. So as much as I could do was just divide the pools, but yeah.
Okay, thank you.
Maybe I missed it, but why did you separate at the very end the trees between northern and southern hemisphere and not, I don't know, tropical or non-tropical?
Yeah, I was looking. We also have the global patterns, and so the global patterns were like extremely strange, basically. So when we started looking at those global patterns, we said maybe it is because it's happening differently in different hemispheres, and started cropping it. So yeah, the full figure has the global, and the global shows you something like that does something like a hump-shaped weird thing. So like, yeah.
Hi, Andrea. Very nice talk. Thanks. Have you looked already to this classification of the Atlantic forest made by Reginato and Michelangeli in South, Central, and North Atlantic forest, by the way?
Yeah,
I would like to see your opinion about this.
Well, I mean, Marcelo and Fabian are part of all of these papers.
Yeah.
So, so we've discussed a little bit about the classifications. But yeah, we think, yeah.
Yeah, just because we are using this classification, three different parts, and see if it's interesting or to divide like this or in two kinds of, as you say, north and south.
Yeah, we could try the classification. We've discussed this many times on how we divide the forest.
Right, thanks.
Thank you. That was a super lovely talk. On your global maps, it just caught my eye because Australia came up as bright red in functional diversity. I'm like, yeah, booyah. But how do you account for the number of trees or the quantity of trees when you're calculating that functional diversity? Because the center of Australia is like, there's one tree every kilometer. So it's pretty diverse. When you're calculating as a proportion of the number of trees, does that impact the calculations? And
yeah, you have to account for the number of traits, or else it just goes gigantic very quickly. So it is corrected by the number of traits. Australia is my biggest question, or our biggest question. I've discussed this many times. I have no good explanation for Australia, and it keeps popping out in different versions of these maps and correcting by different things. It just seems Australia has just a very different evolutionary history, and we just can't account for this red. Yeah.
Interesting. Cool.
And soil diversity, I heard the other day. It just seems.
Yeah, could it be because there's just no trees in the center? How does it deal with desert when it's
I mean, there are some trees, so we don't want to take them out, but we've also looked at just removing the whole desert area, but still the rest of Australia gets its red. I don't know if you're from Australia and have some very good ideas. I really want to understand what's happening there.
Oh, no idea. Sorry, not on that side, but thank you.
Questions, anyone? No? Okay. So thank you for joining us for the last seminar of the semester. We hope to see you here next semester. And thank you very much, Andrea Paz, for this talk. It was amazing. You're welcome to come back anytime.